Hello, everyone, and welcome to our next session, which is on the COVID studies around the world that the IHCC has been involved with. And I would have been Nikki Tiffin for this, except for she is still in Nairobi trying to connect with faulty internet. So I'll go through this real quickly. First of all, let me introduce the panelists. Sarah Baumeister, who I think you heard of in the last session, is a cognitive neuropsychologist and senior scientist at the University of Oxford. She is the senior data manager for the Dementias Platform UK and a co-PI for that pilot project you heard about, global mental health impact of the COVID-19 pandemic. And she will be talking again about her research on the global mental health impact of COVID-19. Michelle Ramsey, who you heard of from very early this morning, is the director of the Sidney Brenner Institute for Molecular Bioscience, professor in human genetics and South African research chair in genomics and bioinformatics of the African populations at the University of Witwatersrand in Johannesburg, South Africa, and president of the International Federation of Human Genetic Societies, as well as, you know, the co-chair of the IHCC. And she'll be talking about the potential impact of genetic diversity on COVID-19 host susceptibility in African populations. Albert Tenesa, who'll be joining us shortly, is a group leader at the Roslin Institute, chair of the quantitative genetics at the University of Edinburgh, where he works on how genetic variation contributes to phenotypic variation of complex traits in humans. He will be telling us about the Corona gene study, where citizen science meets COVID-19 research. And lastly, Ricardo Verdugo will be joining us. Ricardo is the director of the Chile Genomico project, which seeks to characterize the genetic diversity of the Chilean population and its association to health. He also directs the Genomics and Bioinformatics Laboratory and leads the University Genoma National Academic Network to support research and graduate level education in genetics, genomics, and bioinformatics, and the C19 GenoNet multicentric COVID-19 cohort. He is a group leader at the Institute of Interdisciplinary Research at the University of Talca in Talca, Chile. Ricardo will be talking about his research program creating a multi-center COVID-19 cohort for genomic and proteomic research in Chile. Welcome to all our panelists that are here this morning. I hope that you are ready for some questions that Nikki has prepared. And you should be used to this because our webinar that we had last October, we sort of started this and we didn't get through all the questions. So this is a continuation, or part two of that very, very well-received panel. So let me start with the first question. I'll read it out loud and then I'll just go through all of you to answer it. So the first question we have is, how have you approached COVID-19 phenotyping? Let me clarify that a little bit. First, disease susceptibility and disease severity are fairly different phenotypes. And secondly, how have you accounted for ascertainment bias, whereby individuals with SARS-CoV-2 infection and very mild or no systems may not be counted as cases in your study? Ricardo, would you like to start with that, please? Ricardo, you're on mute.
Hello, can you hear me?
Oh, we can hear you now.
Okay.
Your camera's off too, if you want to.
Yeah, apparently my camera stopped working. Oh,
there you go.
Okay, great. Sorry, I'm on my cell phone too. I'll just leave. Okay, so thank you, Scott, for organizing this and helping us. So the question, if I understood correctly, was about ascertainment bias in our studies, correct? So that's actually a problem, but what we can do without designing new platforms for every particular cohort, it's trying to use arrays that have been designed for worldwide populations. And that's what we have done in the COVID-19 genomics network in Chile. We have genotype participants with the global survey array assay by Illumina, which is supposed to capture that genetic diversity across the globe. And we have done different tests with this platform by imputing only with, for example, 1,000 genomes, whole genome sequencing data or adding some local or whole genome sequencing data that we have generated in the Chile Genomico initiative. And we see a lower performance in the imputation for the Chilean genomes than what can be obtained for European genomes when using only 1,000 genomes as a reference. When we add our own genomes, which are mostly almost exclusively Native Americans from southern South America, we do see a significant improvement in imputation. It's not the quality is not as high as for Europeans, but it's over 90% probability of having the right genotype. So it's comparable to other populations. So we think that we are close enough so that we can make use the data. So we are happy with the platform that we selected because of the balance between cost and the quality of the data we get once you get the imputation right. So that will be our approach to this problem.
Thank you, Ricardo. Sarah, would you like to speak on your phenotyping and potential for ascertainment bias in your project?
Yes, so we've not been working, as most people know, with genomic data or genetics, but with our survey data and our clinical self-report data, I think the importance of working with multi-cohort self-report data is the validation work that we've been doing across our studies and going forward, which is, so in the previous presentation, you would have seen our cohort table of 23 cohorts. And a lot of people will say, oh, how can you possibly do an analysis with 23 cohorts?
This meeting is being recorded.
Well, that's not really the aim. The aim is to find comparable cohorts to run an analysis and then to validate that analysis in a comparable cohort. And I think that that is what's really important for us. So if we have one cohort that records COVID status in a certain way, whether it's self-report, have you had COVID? We can't actually say whether that was accurate or not, whether it was a PCA, whether they were really reporting that perfectly accurately. However, if we run a validation analysis in a comparable cohort and we're having similar outcomes, that having COVID twice is associated with an increase in depression, for example, and we're having that similarity across cohorts. So I think for us, that is why it's important for us to have a data corpus of as many collaborating cohorts as possible.
Thank you, Sarah. Michelle, let's turn to you. Phenotyping and potential for ascertainment bias.
Thank you so much, Scott. And I think, similar to Sarah's study, we've also onboarded multiple different studies that were sort of working on their own. So, we tried to put the data together in a new database and to have definitions for asymptomatic, mild, severe. And then, of course, we had those who had died from COVID. So we had positive PCR tests for COVID in our cases. And we have compared this to population controls because we already have those genotyped. So our definition of severe was individuals who had been hospitalized, required oxygen, or were ventilated. And then mild was individuals who had symptoms, and then asymptomatic, no symptoms, of course. So we would clump together the severe and the deaths when we do our analysis. So our GWAS would be between these cases that we've now defined in this way, and then the population controls. So just to follow on what Ricardo was saying, so because this is an African population, we used the H3 Africa genotyping array, and we imputed with the Sanger African panel because that was most appropriate, and we found that to work, best in the studies that we've done. So ascertainment bias is an interesting one. Scott, because we keep on adding to our cohort other studies, because people are now because we've published our paper, they're finding us and they want to, add their cohorts to ours. So it's very difficult, so we indicated that we wanted Black South African. And, we don't have ethnicity or ethnic group on all of the participants. But of course, when we do principal component analysis, we can have a sense of, how well matched are our cases and our controls. So that's quite interesting. But we were targeting or trying through the other studies to get more severe COVID cases. Thank you.
Thank you. Albert, if you unmute yourself, could you answer the question, please?
Yes, so our approach to phenotyping was self-reported to start with, and then linking people to their electronic health records. So that means that during the pandemic, when we were in lockdown, they were reporting their symptoms on the web every day if they wanted to, and then we are now trying to get their electronic health record before they develop the disease and after. In terms of ascertainment bias, I would say that all studies that involve humans have ascertainment bias. This is how much there is and whether it's important to the question that you want to answer. But provided that is volunteer, there has to be some kind of ascertainment bias. Our studies is usually highly educated, especially those that had genotypes, because we ask them to donate their genotypes from direct to consumer genotyping services, and that always means that you have $100 to spare at the very least. So in that sense, that's they are really not representative of the general population. But for the genetics, I'm not quite sure that would have a big effect. For the environmental risk factors, that could have a big effect, I think.
Okay, let me turn to another question for you all. This question is a little bit different. And the question is, how do you manage your informed consent processes with regard to sharing data and sharing data across borders? And Michelle, can I start with you for this question, please?
Yes, certainly, Scott. So that was one of our challenges, was really ensuring that the participants across the different studies that were part of COVID-Gen SA had consented similarly and had given broad consent for future use of both the samples and the data and to trans-border transfer. So we spent a lot of time, putting them all under one ethics umbrella and ensuring that would be possible. So in South Africa, if we want to share, we would have a data transfer agreement with the people that we're sharing with. But we will be able to do so because of the way that we consented and the time that we spent to do that.
Okay, thank you. Albert, any comments on informed consent?
Yes, so our consent also allows us to share data with an MTA, but it's I guess a bit more complicated for us. So we can share the genotype data and the self-reported data. The electronic health record data we can't, and that's because of the NHS that they wouldn't allow us to do that. So it's a mix for us, I guess. Even if the participants consented, I don't think we will be allowed to do that.
Okay. Ricardo, any comments on consent and data sharing?
Yeah, so we designed the informed consent in a way that would allow us to share data, because that was the spirit of the project from the start. And we ask whether you will consent to participate in this study, whether you will consent separately for future studies, whether your sample can be stored in a biobank, and we explicitly indicate in the informed consent that the data will be deposited in repositories, anonymized, of course. So that's what we are going to do. All the data is going to be available. So we have genotypes for around 3,000 participants from Chile, plus some population controls, like 700. And those will be deposited in probably EGA, along with the phenotypes of severity and susceptibility. When that will happen, probably with the first publications. We are working on the first publication. So if everything goes well within the first half of 2023, depending on the revisions that we are asked. But before that, of course, we can share data with MTAs. We have done so already with some groups in Latin America that we are working in pulling together data from different Latin American countries. And if there is people from other continents interested in comparing results or doing meta-analysis or even having access to the raw data, we can do it. It will be in a way of collaboration, and so yes, I think that the whole point for us is to make the more progress and advancing in better understanding this disease. The fastest is the better. So collaboration, it's a great way to move forward fastly.
Great, thank you, Ricardo. And Sarah, consent, informed consent.
So the importance of consent for us, because we're working with all secondary data sets, we found very quickly that each of our data sets had different bespoke models in place. And across the 3 PIs and the 3 sites, each of us were connected to cohorts which we could easily access in situ. So for example, Jordan Smoller with the All of Us program, that was easily accessible on site with the permissions already in place with his analysts. And for Jordan, for Andre Brunoni, likewise for Elsa Brazil. But then when it came to other cohorts in other places, there were problems with sharing the data outside of countries within the time frame that we needed. But going forward, we're setting up a 3-model approach where we have in situ cohorts, the ones which I've named, but also federated analyses approach, where we'll collaborate with cohorts where they will analyze the data. We will set up site contracts with them, and they will analyze the data according to the models that we work with them on. And also we have a third approach where we will use the Dementias Platform UK data portal as a shared infrastructure, as an analyses shared infrastructure. So those cohorts who are willing to upload into a secure repository, much like I think Ricardo was saying about a repository area, then the analysts can access the data in a secure repository and run the same analyses taking place in situ and in the federated. So it's a 3-model approach, taking into account data access requirements of cohorts. So we're not trying to impose on anyone, we're trying to make this approach accessible.
Thank you. Albert, you have your hand up. A question from you?
Yeah, I guess it was more of a comment. I guess sharing data, it depends on what data you share. So for instance, sharing individual genotype data or sequencing data, it's a lot more of an issue than sharing summary statistics. Sharing summary statistics of a GWAS or a risk factor or a FIGWAS or whatever with an MTA, I think it's usually pretty straightforward, and I don't think there is much issues with that. But sharing individual level data that involves genotyping of any sort, that's a big issue. And I think the question should, the answer to the questions is the question is very different depending what you want to share.
Good point. We have a question from the audience that I think pertains to this, and the question is, are there additional regulatory issues in addition to conformed consent? For example, POPI in South Africa, GDPR in Europe. And how do you address those additional regulatory issues? So I guess this may be, Michelle, looking at you and Sarah and Albert, really, if you could comment on that.
Thank you very much, Scott. So it's not an easy sort of environment to navigate. And, we've found, especially in Africa, so many countries don't have regulations in terms of protection of personal information. So it becomes very complex, and it's like a moving sort of landscape. So the additional regulatory things, we always say you have to be legally compliant and ethically sound. And so we work on this, with all our partners all the time to try and navigate that. So, there's ethical oversight, and then there's also the Protection of Personal Information Act in South Africa, which talks about processing and sharing personal information. And that has been a real steep learning curve, for all of us. But I just wanted to make a quick comment on Albert's, sort of saying, what kind of data you share, meaning that that would influence how you share. And individual level data is always, the most sensitive. And we have a lot of our data behind a sort of a managed access, so that we have data access committees. So people don't just openly download the data or share the data. It goes through a process and a committee who will then have a sort of a contract to say what you may and may not do with the data. For instance, you may not share it with a third party without prior consent. Thank you.
Sarah or Albert, any comments on GDPR in Europe and implications for you?
Well, the clearest implication of GDPR for us is that, for instance, we cannot transfer data to the US. We cannot even analyze data in Amazon, for instance, that where the servers might be in the US. So yeah, it has implication, but that I think it's independent of whether the participants have consented or not. That's the law per se. So we actually cannot do it.
I think this is just I refer to my previous answer, this three situation approach that we've taken, that all data for us has to be anonymized, and that by using this secure data repository that happens to be Deep UK because of my connection, it's because they take care of the GDPR and the data security. That is an advantage. It's a bonus for us, but we still can't have certain data sets in Deep UK data portal, from certain countries. We still can't have some American, USA data sets. We still can't have some European data sets. So that's why we've had to draw up this three model approach to data access, and rather than force anything that we wanted.
Okay, let's move on to another question that's less related to consent and data sharing and more about your particular studies. And the question is, to what extent might you be able to adapt the data in your studies for other types of research, or are they all very specific for just COVID-19 research? Sarah, would you like to comment on that first?
I think that what we attempted to do was, step one for us was building the collaborations. And I think we had a very minimal requirement for collaboration. So we were looking for cohorts initially that had socio-demographic data and that had collected mental health pre-pandemic and during the pandemic. And I would say, off the top of my head, most cohorts have collected some mental health measure, and definitely socio-demographic. So I think that has been the success to us having over 23 cohorts at the moment in our data corpus. And so where we go from there and where we take our scientific questions, maybe we use only 3 of those cohorts for one question, 6 of them for another. We have built trust with a collaboration of a large group of cohorts, which doesn't exclude expanding our research question. So we could extend that to well-being, we could extend that to other research questions. But because we have worked so hard, I think that most of our pilot study work was on building that collaboration. And I think in a way it was a bonus that our research team worked so hard to get 4 publications in the research question. That is a bonus and a credit to them. But I think that the open approach in the beginning will pay off to being to creating this open environment.
Great. Albert, any comments on other uses of your study?
Yeah, we have broad consent, so potentially we could test hypotheses that relate genetics to disease or future diseases with COVID or pre-COVID diseases, and potentially we could do that. Yeah.
How about
I
you?
think yeah, I think usually researchers tend to be as broad as they can, because if you are too restrictive, then you never know where the data is going to take you, and if you are very restricted, then you may end up in a situation where you have a very good hypothesis, and you have the data, but you cannot test it, and that might be very frustrating. Well, it would be very frustrating.
Ricardo, any thoughts on other uses of the data you've collected as part of
Yeah,
your COVID studies?
In our case, the cohort was started with the pandemic. We don't have data, previous data or data of people that was not infected. So both by design and by informed consent restriction, we have to study COVID, but also other pathologies that may be triggered or related indirectly with COVID. So we collected information about over 20 different diseases, chronic diseases that the participants may have had a diagnosis for before the pandemic and after the pandemic. And we are now in the process of recontacting every participant. Well, actually, we already just finished that. And so we asked them again whether they have new diagnosis of many other diseases on top of the usual signs of long COVID. So people interested in studying mental disorders or metabolic diseases, we have the data for that, and we plan to continue collecting that data prospectively. So I think that there is room for doing studies that are not only focused on COVID, but also the relationship between COVID and triggering other or favoring other diseases as well.
And Michelle, this may be the last response to the question we have. And I also remember a conversation you and I had recently about cohort of cohorts collaboration, where we looked across IHCC. And I wonder if the 4 panelists right here could form the nexus for that sort of study with what we just heard about, the types of data they've collected and the potential for other applications.
Thanks, Scott. I think you 100% right. And, I'm also with Albert and Ricardo about, being able to go back to participants, because we're also collecting longitudinal data. And one of the sub-studies that we're going to do now is to look at non-seroconverters for SARS-CoV-2. And we're going to do whole genome sequencing on 30 individuals. And, one of the other uses is that, of course, African data is very scarce. So we could use those whole genomes to be part of an imputation panel that we are developing for African populations. So I think certainly opportunities to do population genetics, other studies with this data, which isn't part of the original objective, but because of the broad consent and being able to go back to the ethics committees to say this data would be useful for these purposes, that would be a possibility. So very exciting to think about what we may do with this data. Thank you.
Okay, well, I just want to finish up by thanking all of you. Your studies have been amazing. We've heard about them in some of the pilot projects and sessions earlier today. There's more information available on the website. And I also thank participants that have sent in questions. We'll try and answer those offline. And particularly for those of you that have foregone the first half of the World Cup games this morning for joining the sessions instead, I'm glad that you were here and that you can now go catch the second half of those games, perhaps a little bit later, maybe even a little bit at the end of the first half. But thank you all again for a wonderful session. We greatly appreciate it.